So, here's the scoop. We covered all the bacterial structures, and you'll need to know them. The one we didn't cover is a pilus and a plasmid.
This is good.
And they actually cover it in chapter 7, genetics. But I don't want to wait till then. So we're just going to do it briefly. Like I said, I'm not a fan of the way this book is organized. But okay, here we go. Here's one bacterial cell. Oh, somebody cleaned the board nice. Here's a bacterial cell. I made it a nice bacillus-shaped cell. Doesn't have to be bacillus-shaped. Here's its nucleoid, plasma membrane, cell wall, cytosol inside, couple ribosomes, whatever. Might have a outer gooey capsule, might have flagella, might have fimbria, fingers for attaching. But what we're going to focus on is a plasmid first. So let me use a different color for the plasmid so that it's easy to see. I'll use red. Okay, that's the nucleoid. This is the plasmid. Plasmid is not a species trait. Every member of the species might not have it. But if you have it, you might be quite special. Because if you have a plasmid, the genes on a plasmid might enhance your virulence, which means the symptoms for the patient are going to be worse, but it's better for the bacterium because it helps them colonize and infect the patient. One of the things that might be on a plasmid is a gene for antibiotic resistance. It might be a gene for a special protein or enzyme. Maybe a toxin. And another gene that's on the plasmid is a gene for the pilus. Pili would be plural. Now, they don't talk about this particular thing until chapter 7, the chapter on genetics. But I also have it in your handouts package under gene transfer mechanisms. So, we're still talking about structures of bacteria because these structures are some of the structures you're going to need to know. If a bacterium has a plasmid, it also has a pilus. And a pilus is a hollow tube through which it can make a copy of its plasmid. So it'll open the plasmid, copy it. You got a little copy. The copy's going to go through the pilus, and where do you think it's going to go? To another bacterial cell. And it doesn't even have to be the same kind of cell. It can be a member of a different species, which blows your mind if you're a traditional biologist, because in traditional biology, what makes a species a species? Colton. You can procreate. You can mate with members of your own species successfully. But if you're two distinctly different species, you cannot have successful reproduction. Here's two different species of bacteria, and yet they're doing conjugation. It's not replication, however. It's just conjugation. So, also they have this documentary this summer they've been running on WEDU, the public broadcasting station, called First Peoples, and about how that's all changed, how we think we evolved, and that we actually have Neanderthal genes in us. Because modern humans, as modern humans started to evolve, we actually ran into Neanderthals and mated with them. So we actually have some Neanderthal genes. So when people call you a Neanderthal, they're probably right. Yeah. And apparently, it's only Europeans who did this. Other humans that went to, say, Australia or whatever didn't meet Neanderthals, so they don't have Neanderthal genes. But see, they can do all this stuff because now they can sequence the DNA. So if they can find a fossilized bone of a human from 40,000 years ago and actually get some DNA out of a tooth or a piece of the bone, then they can compare it to other DNA of Neanderthals and all whatever. Never mind. Anyway, so here's another bacterial cell, cell wall, plasma membrane, nucleoid, ribosomes, whatever. And now it gets a copy of the plasmid, right? Curls up inside, and it's got the plasmid inside. So, when this cell started out, it was considered F+, like male. It has the thing, okay? This one, before conjugation, she did not have a plasmid, so she would not have a thing. So she was considered F-, that's like female. Once he gives her a copy of his plasmid, the gene for the pilus is on the plasmid, she grows a pilus. Now just think about that, ladies. Did you ever wonder how it felt to be on the giving end? And now she is a he and can conjugate with the next cell. And this is one way that bacterial populations transfer antibiotic resistance. Or perhaps they transfer necrotizing fasciitis enzymes. Or perhaps they transfer a toxin, an erythrogenic toxin or something like that. So this is how a population of bacteria can actually change. If a few of the individuals have a plasmid and they can pass copies of it to the other members in the population, that's how antibiotic resistance is thought to have developed so quickly. Not just by mutation, that wouldn't have happened fast enough, but by these other mechanisms called gene transfer mechanisms. So gene transfer mechanisms, remember we already talked about this a little bit, and Anita answered our question. What do gene transfer mechanisms give bacteria? Genetic recombination, new genes. Essentially. This particular gene transfer mechanism is called conjugation. This is not happening during transverse binary fission. It's a separate event. And you'll have that question on your test, because I wrote the same question like 5 different ways. So depending on which copy of the test you get, you'll get a question about transverse binary fission. In transverse binary fission, one cell copies its nucleoid, elongates, splits in half by forming a transverse plate or cross wall, and then the 2 cells separate into individual daughter cells. That's replication. One parent cell becomes 2 daughter cells. That's replication or reproduction. This is genetic recombination. And you're going to have 2 cells involved here. Okay, there's other gene transfer mechanisms. They're also in your handouts package. Perhaps you remember what they were. Transformation and transduction. We talked about those a little bit. They're in your handouts package. They'll come up later in a PowerPoint for Chapter 7 on genetics. But right now I wanted to mention conjugation because I'm trying to explain the bacterial structures of plasmid and pilus. So this is the pilus. Okay, so we finished talking about bacterial shapes and arrangements. and bacterial structures. So your second test will have all those on it. Not the one you're taking this weekend, the next one. And I borrowed somebody's book. But if you like to read ahead, it's in chapter 7. The next chapter where you can use the PowerPoint. Alrighty. Did that. Alrighty. Check. Did bacterial structures. Now we got to do the cell wall. So the cell wall is actually the last bacterial structure we're going to do. And it coincides with classification and how to do your bacteria list. So we'll start on that. And I'll just read you a cute little story before I forget. We talked about penicillin, and Fleming discovered penicillin. Well, somebody, Felix Risk, who used to work at the Venice campus, he died. It was so sad. He had two boys and a girl. He died of cancer. Anyway, he sent this to me a couple of years ago, before he died, obviously. And it's a story. And it goes like this. His name was Fleming, and he was a poor Scottish farmer. One day, while trying to make a living for his family, he heard a cry for help coming from a nearby bog. You know what a bog is? Like a cranberry bog you see on the TV commercials for the cranberry juice. It's like a big pond, and it's filled with mud and muck and like a nasty, mucky pond, like quicksand almost. Farmer Fleming dropped his tools and ran to the bog. There, stuck in the black muck up to his waist, was a terrified boy. The boy was screaming and struggling to free himself.